Namaskar. Today we'll study cardiac muscle contraction mechanism. And cardiac muscle, you can see here the structure of a sarcomere, where in the center there is thick filament, and each thick filament is separated by a thin filament. So on both sides of the thick filament there are two thin filaments, and these thin filaments form the jet disc. And this thick filament consists of myosin, and this is the structure of myosin. This is myosin tail and this is myosin head, and these myosin heads are projected here, and the tails form the thick filament. The thin filament consists of the contractile protein actin, and you can see here this is the structure of actin. So this is the thin filament, and this red part is the myosin binding sites of the actin. This is actin, and this is the myosin binding sites of the actin. So myosin head can bind to, suppose this is the thin filament, and this is like this. So here the myosin actin is there, and this is the myosin binding site, and when this myosin head binds to this one, this will pull. So the direction will be like this here and in this direction, and there will be contraction of this muscle. So we call it as the slide filament theory, and this is the same mechanism as that of the skeletal muscles. And because of this sliding of the thin filaments over the thick filament, there will be contraction. And how this occurs? The myosin heads will bind to the actins, and these thin filaments will slide towards the thick filaments. That will result in the contraction of the skeletal muscle as well as cardiac muscle. So we are discussing today on the cardiac muscles. So actin myosin binding results in sliding of the thin filament over the thick filament, and there will be contraction of the cardiac muscle. And these myosin binding sites of actin are covered by another layer called tropomyosin. So tropomyosin covers the myosin binding sites of actin, and that tropomyosin is regulated by another protein called troponin. So two regulator proteins are there, troponin and tropomyosin, and two contractile proteins are there, myosin and actin. So actin myosin binding is regulated by troponin and tropomyosin. And how it occurs? Calcium has the affinity to bind with this troponin. When calcium binds to troponin, troponin replaces tropomyosin. The myosin binding sites of actin are exposed. Then that will bind with the myosin heads, and there will be movement of the thin filament towards the thick filament. Sliding filament will be there. Sliding of the thin filaments towards the thick filament will occur. The jet discs will move towards each other, and there will be contraction of the cardiac muscle. So in heart we have the beta 1 receptor, which is a GS type G protein coupled receptor, which is linked to the effector adenylate cyclase, where the second messenger is cyclic AMP. ATP is converted to cyclic AMP in presence of active adenylate cyclase. Cyclic AMP activates protein kinase A, which binds to the ryanodine receptors to release calcium, and now this calcium binds with troponin. Protein kinase A can also activate troponin, and troponin alters the tropomyosin, and the myosin binding sites are exposed. Actin myosin binding occurs, which results in contraction of cardiac muscle. So this is all about the physiology or mechanism of cardiac muscle contraction. Thank you.